Hey everyone, we're going to be talking about macromolecules, the building blocks of life. When I'm talking about macromolecules, I'm talking about big molecules because macro means big. They come in 4 major classes that we will take a look at each one separately. Carbohydrates, lipids, which some people also call fats, and proteins, and nucleic acids, also known as DNA and RNA and ATP. They all build our bodies. But before we talk about those specifically, we're going to talk about what they're built out of and what they have in common in the way that they are built or taken apart. All right, so the word polymer, take a look at that. Poly, the prefix, means many. And so in this case, some of these macromolecules are classified as polymers. It means they're made out of repeating units called monomers. Mono means one mer. All right, so a polymer has many monomers, repeating units. Like, I'll just give you an example right now. Proteins are made out of repeating units or monomers called amino acids. Carbohydrates, many times their repeating unit or monomer is glucose. The only one that is not a polymer is lipids. Lipids are actually not considered a polymer, but the way they're built and the way they're taken apart, they all have that in common. So we'll look at that first. One thing, we've got stable, nice covalent bonds that build these things. Remember, those are our strongest bond. And then the way that we build them is called a dehydration synthesis. So we'll learn this once. This dehydration synthesis applies to all 4 of the macromolecules. This is how we build them, hence the word synthesis. But you also see the word dehydration in there. Dehydration, remember, you're dehydrated, you're lacking water. Well, it's the same thing here. It's when we take water out in order to build our polymer, whether it be carbs or proteins or nucleic acids, or, well, lipids are not a polymer, but we build them with the dehydration synthesis as well. All right, so each of these green circles then we'll pretend is a monomer. And we have some other things hanging off the end. Each one of these has an H and an OH on opposite sides hanging off. And so what happens is when if I can click here, there we go. If there it is, that's what I was looking for, the enzyme. An enzyme grabs onto this H and this OH and then takes it off. So when you have an H and an OH, what's the result? Well, it's water. And so that's where the word dehydration comes from. So if I remove the H and the OH, basically that creates a bonding site for our 2 monomers. Maybe these are 2 molecules of glucose connecting, or maybe they're 2 amino acids that are connecting. Enzymes are proteins that do specific jobs in the body. So there are specific enzymes that complete the dehydration synthesis process. All right. This does require energy as well as the enzymes. Sometimes it's also called a condensation reaction, but 99.9% of the time I call it a dehydration synthesis. All right, the opposite, that was building, but this, a digestion, is breaking it down. Sometimes we have to break macromolecules down, like when you eat a big meal of carbohydrates, proteins, and lipids. You've got to break those down into their individual parts or individual monomers. Then later you'll build them back up into the ones that make you or the ones that you need. All right, so in this case, remember last time we took water out? Well, to do a digestion or to tear this thing apart, we're going to put the water back. All right, so we're going to have an enzyme come along here and it will put some stress on these bonds right in between these two monomers. Put stress there and it'll break that bond. But now I've got two open sites that I've got to fill with atoms. So on one of the sites I fill it with a OH, and the other site I fill it with the H. So I had to take water apart and put it back into the molecules in order to tear this thing down. Yes, it's called a digestion. Most of the time I also call it a hydrolysis. Number one, because I know that the suffix lysis in science means tearing apart or destruction or breaking down. Like later on we'll talk about cell lysis, and it means tearing down of cells. Hydro reminds me that I'm putting the water back in. So I like that word best. I like hydrolysis. It tells me I'm putting the water back in, and the lysis part tells me that I'm breaking it apart. This type of reaction releases energy. Alright, so now that we've talked about a little bit more about how those macromolecules are built and taken apart, and the fact that they're made out of repeating units called monomers, let's talk about carbohydrates specifically. Carbohydrates, very familiar, very yummy and tasty. Some of them taste sweeter than others, but most of us love our carbohydrates. Oh yeah. They are nicknamed our energy molecule. And the carbohydrate that we see right up here in the upper right corner, that's glucose. A building block or a monomer of many carbohydrates. There was a fad, and probably still is in many places, of low-carb diets because they add sugars like glucose to your system. People try to cut carbohydrates out of their diet to reduce sugar and therefore lose weight. It's kind of hard to do because they're so delicious. Take a look at the word carbohydrate. It has carbohydrate. Basically, it means this, that it contains a carbon, and a hydrogen, and an oxygen. And then the hydrogen to oxygen ratio is usually 2 to 1. Like in glucose, C6H12O6, the 12 to the 6 is a 2 to 1 ratio. Or sucrose, C12H22O11, that's also a 2 to 1 ratio. A main job for them is energy. And there is some building materials that they're used for, and energy storage as well, like when we store carbohydrates as glycogen in our muscle and liver cells, or how plants store starch, which is a carbohydrate, in their cells. Their monomers are sugars, and it's usually glucose. And remember, carbohydrates are classified as a polymer. So sugars, starches, and cellulose, that's the type of sugar that makes up the cell walls of plants. Alright, if you want to know if something's a sugar or not, look for the ose ending, like glucose, fructose, and sucrose, and lactose. All of them end in an ose, and even cellulose. Some here that we see are a 6-carbon sugar like glucose, and some are a 5-carbon sugar like ribose. When it's a 6-carbon sugar and you put it in water, it curls up into a hexagon. And when it's a 5-carbon sugar in water, it curls up to be a pentagon. Glucose is kind of the universal energy sugar. Ribose, we use that in RNA. That's the sugar that's in RNA. Then there's another 3-carbon sugar that's just a straight linear model. And that one is glyceraldehyde. And it's just linear and straight. Oh, notice we've got a carbonyl ending on this one. And here's some examples. They have the carbonyl endings. Sometimes it's on the end, and if that's the case, it's an aldose. If it's a carbonyl in the middle of the structure, then it's a ketose. Here's glucose in its linear form. Put in water, then it forms a ring. A hexagon, actually. So where do we find solutions in biology? Well, in our body, we have 70% water, so these sugars are in solution within our cells. And the carbons are numbered. This is going to be important later on. All right, remember when glucose forms a hexagon that we don't even have to draw the C's. We just assume they're at each corner, but we count them. So this is the 1' carbon, the 2'. The 3' has an arrow at it because that is going to be such an important carbon when we talk about DNA and RNA. There's the 4, here's the 5. That one gets a green arrow as well because that carbon is going to be so important when we talk about DNA and RNA. This 3' and 5' carbon kind of help us know which direction we're going in DNA and RNA. And then the other carbon sits above it. So also remember that with these carbon bonds, there's potential energy stored in those bonds. In fact, when you do cell respiration, you break down glucose to harness that potential energy by breaking down those bonds. But we'll get to that in a later chapter. And in classifying carbohydrates, if it's made out of just one simple sugar, it's called a monosaccharide, one monomer. And an example of a monosaccharide is glucose, and fructose is another example. Then disaccharides contain 2 simple sugars or 2 monomers. An example would be sucrose, which is plain old white table sugar. Lactose, which is milk sugar, is another example of a disaccharide. And maltose, which is often found in malt beverages like beer. Poly means many. So as you can probably guess, this is when we chain a lot of monomers together and we make these long chains. Hence the word, it's a macromolecule. And they become very large polymers. So starch and cellulose, which we find in plants, those are polysaccharides. And in humans, we would have glycogen, though I don't have that one pictured here, would be a polysaccharide. So in building sugars, let's go over this again. We've got 2 glucoses here. And I'm going to start building a carbohydrate, a polymer. So in order to do that, I'm going to have to join these 2 glucoses together. So if you take a look, we've got an OH hanging off one and this H over here. If we remove that, then we create a bonding site where they can join together. But in doing so, in removing the H and the OH, we remove water. Hence, remember the term dehydration synthesis. These 2 monosaccharides are now joined to make a disaccharide. The disaccharide we're looking at here, when we join 2 glucoses together, is called a maltose. And we find that often in malt beverages like beer. It's not so sweet, basically. When you join 2 sugars together in a dehydration synthesis, it's called the glycosidic linkage. The prefix gly means sugar. So we've linked 2 sugars together. Let's try another one. Here we've got glucose and fructose, both monomers or monosaccharides. We'll do the dehydration synthesis again and remove water so that we can create the glycosidic linkage between them. And what do we make? This is how we make table sugar. Just plain old white table sugar. This type of sugar and fructose we find in fruits and candies and table sugar. So again, a monosaccharide is one monomer, one simple sugar. Disaccharide is two monomers linked together. And a polysaccharide is when there's many. It costs a little energy to build these, actually. The ease of the dehydration synthesis. It's easily reversible, relatively quick. So the function then in plants is storing polysaccharides, storing basically chains of glucose as starch. We store chains of sugars or chains of glucose as glycogen in our liver and muscles. Now, starch and glycogen are very similar in that they are made of chains of glucose, but the way they're linked are a little bit different. We'll look at that. And then there's also one that's more for structure, which is cellulose. And cellulose is basically the polysaccharide that makes up the cell walls. We actually do not have a digestive enzyme in our guts to digest cellulose. So to us, whenever we eat plants, there's plenty of fiber, which is the cellulose, that we can't digest, which helps to keep our digestive system regular. Another example of a polysaccharide is one called chitin. It's in arthropods and fungi. The arthropods, it's in its exoskeleton. Again, we don't eat that part. Like when you eat lobster, you remove the exoskeleton. It's the chitin because you don't have an enzyme to digest that. And they make up the structure. Whereas the starch and glycogen is more for the energy storage. It's a good, quick way to store energy. So here's what they look like. The little green hexagons, those are all chains of glucose. So this is what starch looks like. You don't have to memorize what it looks like, just that it's chains of glucose. It's fine with me. And glycogen looks very similar as well. Chains of glucose, long chains of glucose. And we store those in our liver and muscle cells, and we can quickly break glycogen down back into glucose again so that we can quickly use it and break it down so that our mitochondria can make ATP. So again, these are both energy storage. The starch is more of a slow release, but the glycogen for animals is more of a fast release. Just the way the bonds are put together and the structure that it ends up being. All right. So, glucose can actually come in different forms. This one is called alpha glucose. This one's called beta glucose. Really, the only difference is the location of the hydroxyl group. Here it's on the lower end. On this one, it's on the higher end. Because of that location, it's going to kind of make the bonds have a different shape, so to speak. So remember, these are isomers of each other. They have the same formula, but a little bit different structure. And structure determines function. All right, the alpha glucose is the one in starch. The beta glucose is the one in cellulose. And this is going to be an indicator of why we can and cannot digest either one of these here. So the starch, the enzymes that we have can actually break that down. But, oh, nope, it's not working here for the cellulose. If I do that again, nope. We don't have an enzyme. Take a look at the structure here, having that beta glucose. Every other bond is up in the air. And we just don't have anything to deal with that. You have to have a molecule to fit the shape of this, and we just do not have that. So we can easily digest starch, but not cellulose. But guess who can? Cows. Even though I shouldn't say easy, it's hard for them to do it as well. It's too bad, because cellulose is maybe the most abundant organic compound on Earth, And if we could digest cellulose, we would have plenty of glucose whenever we wanted. But if we can't digest it, it's basically only fiber for us. It'll go right through us. But herbivores have evolved a mechanism. One of those mechanisms is to have guts full of bacteria that can help them digest it. Carnivores, we don't have that. So we call cellulose undigestible roughage. Basically, it pulls water into your large intestine and makes you go number 2 more easily. So it's good to eat cellulose every day. But in a cow here, they have quite the intricate digestive system with different chambers of the stomach, and they even regurgitate if you follow the arrows of the food going in, then it goes through some of the digestive system, then they regurgitate, and they have to re-chew it. Yum, huh? They re-chew it, re-swallow. They've got bacteria helping them out, and finally, even after the fact, after they poop it out, there's still nutrients left that they couldn't digest. But at least they got to start at it. So these types of organisms, like these little moths or butterflies, they're called coprophages. They like to eat the poop because there's a lot of nutrients, the cellulose getting broken down, that are left in that. Yum, huh? Gorillas, they can't digest it well, maybe a little better than us. So they have to supplement. They do eat leaves and stuff, but they supplement with other fruits for their diet. Oh my goodness, who is that? Looks like some delicious carbohydrates there. Let's see, can you recognize that cute little girl there? Huh. She's going to eat some carbohydrates. All right. Eat those carbohydrates. All right. Well, or maybe we should build some. Let's see.